So, ladies and gentlemen, you are all welcome to my YouTube channel. So, today we are going to look at the lipid absorption. So, generally, after the digestion of the lipids in the small intestine by three major enzymes, pancreatic lipase, cholesterol esterase, and phospholipase A2. So, when these three major enzymes in the small intestine hydrolyze the lipids, so we have, or usually there are there are products generated, like for example, when the enzymes that work on phospholipid is phospholipase A2, and once these enzymes work on phospholipids, a lysophospholipid is generated, and this lysophospholipid is one of the major emulsifier. It's one of the major emulsifier that use in emulsifying detergent. So a lysophospholipid is actually generated when these enzymes act on the phospholipid, and of course, when for the triacylglycerol, an enzyme called pancreatic lipases break down triacylglycerol, that is TAG, triacylglycerol, into free fatty acids and glycerol, So a long chain free fatty acid and glycerol are generated when an enzyme called pancreatic lipase hydrolyzes triacylglycerol. And of course, an enzyme called cholesterol esterase is also another enzyme in the small intestine that degrade cholesterol ester into cholesterol with one long chain fatty acid So it means that at the end, after the action of these three enzymes, phospholipase A2, cholesterol esterase, and pancreatic lipases, there are products that are generated. And these products that are generated include lysophospholipids, monoacylglycerol, long-chain fatty acid, and cholesterol. But these products generated cannot cross the membrane of the intestine. So what will happen? They need a carrier that will actually emulsify them to be able to cross the membrane. So that is why, as a result of that, you need a bile salt. And this bile salt, combined with all these products of digested products, lysophospholipids, monoacylglycerol, long-chain fatty acid, and cholesterol. So when all they combined with the bile salt, will form what we call a mixed micelle. And the nature of the mixed micelle is that all the non-polar component will remain hidden. That is, the hydrophobic component will remain hidden, while that will be at the core of the micelle, while the polar component will be at the surface. So it means that in this surface, all this surface is made of a polar group. It's made of a polar group that can be dissolved or that can be associated with an aqueous medium. So the major aims of forming this mixed micelle is to make these digested products into a molecules that can be easily passed across the membrane of the intestine. So as a result of that, these bile acids will now mix with all the digested products to form the mixed micelle. And this is the mixed micelle, and the nature of the mixed micelle is that all the polar component will be on the surface, and then the non-polar component will remain in the core, that is, will be hidden, so that at least it will not be exposed to the aqueous environment. So when you have this, then the next thing is, after you have the micelle formation, so this is actually the mechanistic or the mechanisms of the lipid absorption. First, the bile acids react with all the digested products to form a small micelles, that is the mixed micelles. And then these mixed micelles can be associated with the aqueous medium, and then will now pass through, or it will now be absorbed through the villi and the microvilli. And of course, after they pass that, then they will now combine with what you call a chylomicrons, which is a major transporter that will now carry these end products and then transport them into the lymphatic system. So generally, when lipid is digested and then absorbed, it is usually absorbed and then transported into the lymphatic system. So now let's look at what happened for this lipid absorption. So in the small intestine, the products from fat digestion and bile acid will form a micelle, which moves toward the microvilli to allow the lipid to diffuse into the mucosal cells. So this is the bile acid and then the digested lipids, then they will now produce a mixed micelle, all in the lumen of the small intestine. And then of course, the fatty acids and then the digested products will now pass through the microvilli. So after they pass to the microvilli into the mucosal cell, so inside the mucosal cell, the fatty acid and the monoacyl glycerol and then resemble itself to produce another triacyl glycerol and then incorporated into the lipid transport particles called chylomicrons. So chylomicrons is the transport particles that incorporated with the re-esterified fatty acid, and then it will now take it and transport it into lymphatic or into lymph vessel. So the small intestine is very efficient in absorbing fats. So very little is normally excreted in the feces. So now, in addition, we should know that in the lipid we have short chain fatty acid and a long chain fatty acid. So for the short chain fatty acids, it will not require any transport. They can just easily diffuse into the microvilli and then will now transport it into the blood circulation using albumin. So ladies and gentlemen, this is a little video on how the lipids are absorbed in the body system. And if this is the first time you are coming to my YouTube channel, please subscribe. Thank you.